Hello, everybody. Dr. Vasquez here, coming to you live, and we're going to talk about part 5, what I'm going to call part 5 right now of B12 pharmacology. And this is going to be pretty fast, I hope. So what you've seen me do in the first 4 parts of this is you've seen me kind of build this up, and kind of construct an accurate model, but also to a large extent somewhat a new model, at least in my opinion. Now, or maybe I should say it's an established model. I mean, which is, most of it's just biochemistry, but I have kind of a different interpretation of that biochemistry. So that's the version that you've seen previously, the one that just disappeared. And then this is the new version that I just finished this morning. So, like I said, what I want to do right now is just go through this rather quickly and show you what's pretty close to the final version of this. So, again, I'm calling this B12 Pharmacology Part 5, and I think I'll do a Part 6 just with a few more details, perhaps even as solely a written document. I don't know if I'll do a video on that. But we're pretty close to the end of this conversation. So let's review this final version of the diagram, which again, I just finished over the last couple of days, including this morning. So vitamin B12, we consume vitamin B12 from the diet, animal foods. The classic best example, of course, is beef liver, because beef liver is the most kind of concentrated, one of the most concentrated food sources, period, for iron, for vitamins, lipids, vitamin A, for example, as well. So we consume vitamin B12 in the diet. The other name for B12 is cobalamin. So the name implies that this is a cobalt containing amino acid, basically. Again, we get this from diet, supplements, or injections, either intramuscular, intravenous, or subcutaneous. Holotranscobalamin is the transcobalamin transport protein, which accounts for 10 to 30% of B12 bioavailable. Well, let's see. It's 10 to 30% of the B12 in the blood, but it's 100% of the bioavailable B12. So in order to enter the cell, the vitamin B12 has to be in this form. It has to be transcobalamin protein, which is the transport protein, with B12 connected to it, so that it can then enter through this receptor that I've diagrammed right here. So when we say holotranscobalamin, we're talking about the transcobalamin transport protein with B12 connected to it. That gets endocytized here in this cell membrane receptor called the transcobalamin receptor. So anytime, most receptors are mostly protein. Anytime we have a transport protein or an enzyme, those are gene products, and anytime we have a gene product, the gene can be mutated, and if the gene is mutated, then the product is going to be mutated as well. So all of these that you see in green, you see green here, I'll go through these in more detail in just a moment. All of these that are in green, these are proteins, either transport proteins and or enzymes. Some of these, like CBLC that I have highlighted and bolded right here, it's a transport protein, but it has a little bit of enzymatic action to it because it actually changes the molecular structure of the vitamin B12. So I had some debate in my own mind, and I reached out to some famous experts to talk about this and get some clarity. But the clarity that I have now is that these are transport proteins, and that was a little bit obvious from the research. But the part that I was not clear on for a while was, how can they be transport proteins and function as enzymes at the same time? And that's basically the truth. So you could think of these as being 80% transport proteins and 20% enzymes. So they have a little bit of enzymatic action, even though they're mostly transporting and directing B12 through the cytoplasm. So again, we went from the serum here we have the B12 bound to the transcobalamin protein. It gets endocytized through the receptor here. It enters into the cell again as B12 with the transcobalamin protein. This enters into the lysozyme, where the lysozyme degrades the protein that is the transcobalamin transport protein, and then B12 gets spit out the other side. So we're going to enter into a lysosome here, digest the protein, and then extrude the B12 moiety, so to speak, or the B12 vitamin on the other side. At this point, we're going to remove whatever is attached to that B12. So as you know, B12 exists in several different forms. It could be the synthetic cyanocobalamin, it could be hydroxocobalamin, it could be adenosylcobalamin or methylcobalamin. All those functional additional groups are going to get ripped off of the molecule at this stage. So this CBLC protein has another name called MMA-CHC. This functions in the cytoplasm as a transport protein, but it also removes, in an enzymatic manner, it removes the additive groups that are on the B12 molecule. Again, that could be cyanocobalamin, it could be hydroxocobalamin, methylcobalamin, or adenosylcobalamin. So regardless of what you consumed, at this point it is purely B12 without any functional groups on it. So again, we came from the diet, we came from injections, whether that's intramuscular, subcutaneous, or intravenous. The B12 got bound to the transcobalamin protein, it entered the cell and then entered the lysozyme, and then the B12 was removed from the transcobalamin protein, and then it gets transported by these other proteins that have a little bit of enzymatic activity to them, we might say. So CBLF, CBLJ, anytime you look at a gene product that's a protein or a protein that functions either for transport or as an enzyme, then that protein can be mutated. So some people have mutations here, some people have mutations here, and they can't push the vitamin B12 down the chain of events that we're seeing represented here in my diagram. The most common defect in all of these pathways is this one right here called CBLC. This is the most common genetic defect in B12 metabolism. But some people don't make this receptor, for example, or they have problems with the lysozyme, or they have problems with either of these proteins, or either of these proteins, or either of these proteins. So here's what I just wrote this morning. All proteins and enzymes are subject to mutation and therefore subject to dysfunction. So CBLF can have multiple mutations. CBLJ can have multiple mutations, CBLD and CBLC. So when I've been reading the research on this, what I found is that in any one of these proteins, it's not just one type of mutation. It could be various types of mutations. And so each case is quite different and quite unique. Not all of these are the same. Not all of these are the same. So let's assume that we have B12 in the blood. The B12 enters the cell. It goes through the lysosomal digestion, so to speak, or processing, gets transported here. All the functional groups are removed. And then we're going to take this route through CBLD, CBLE, and CBLG to form methylcobalamin, which is the cofactor required for the enzyme methionine synthase. And that functions here with the folate cycle or with the methionine cycle here. So a few days ago, I introduced the topic of, or the concept of metabolic detoxification. You have to have this pathway functioning in order to get rid of homocysteine. So homocysteine, as I said before, is neurotoxic and vasculotoxic, and it also promotes osteoporosis. So I guess we could say that it's osteotoxic. So you have to have a way to detoxify homocysteine. And I also mentioned, typically when we talk about detoxification, we're talking about chemicals and drugs and xenobiotics and heavy metals. That's typically a phase 1, phase 2 oxidation conjugation reaction followed by excretion, large molecules through the bile, small molecules through the urine, also through lungs and through the skin in the form of sweat. So this is distinct from that. I call this metabolic detoxification, different from xenobiotic detoxification. So I'll talk more about that another day. So again, this is the entry into the cell, the processing within the cell. Any one of these pathways can have problems and mutations and lead to accumulation of these neurotoxic, vasculotoxic, or osteotoxic molecules. In this case, we're picking on homocysteine. Defects on this side lead to defects in DNA, metabolism, and function. And so this is why we see megaloblastic anemia, for example, in people who are B12 and or folate deficient. This also explains a little bit, even though it's not highly detailed, of course, but this also explains a little bit how you can compensate for B12 deficiency with high-dose folic acid. So, one of the concerns that we've had now for decades is the concern that someone could be B12 deficient, but if they're supplementing or even consuming fortified foods that contain folic acid, then that would compensate, at least partly, for the B12 deficiency, and it would mask the B12 deficiency by kind of compensating and covering what would otherwise be a megaloblastic anemia. And you can kind of see that represented here. You can imagine, at least a little bit, that if we had a defect here, we might be able to compensate for it with a little extra folic acid. So again, that's half of the pathway. We're in the cytoplasm here. The B12 went from the blood, connected to its transport protein, entered the cell, and got processed into this route to create methylcobalamin. Methylcobalamin functions in the folate cycle and the methionine cycle. If we don't have this part of the equation working because of lack of B12, lack of folic acid, lack of B6, then we're going to accumulate homocysteine, and that is toxic to the brain, the vasculature, the kidneys, and the bones. So, again, this is half of the equation. I'm going to read this again. All proteins and enzymes are subject, those are gene products, they are subject to mutation and therefore subject to dysfunction. So some people have a defect here and they die without appropriate intervention. The appropriate intervention most commonly is high-dose hydroxycobalamin to basically force this pathway to work even if it's a little defective. And a lot of times that works and sometimes it doesn't. And sometimes it works a little bit, but it doesn't work completely. So again, the most common remedy for defects in this pathway of B12 is to give high-dose hydroxycobalamin, either orally, 2,000 to 6,000 micrograms per day, or by injection, either intravenous, intramuscular, or subcutaneous. In some situations, administering either methylcobalamin or adenosylcobalamin might have some benefit. I'll talk about that more in just a second when I talk about adenosylcobalamin, which gets formed in the mitochondria. So again, you have to assume this is the cell, and obviously it doesn't include everything, but this is the cell. Here, we're in the cytoplasm, and in just a moment, we're going to be in the mitochondria. Before I move on, let me read what I wrote here. MMA-CHC, also known as CBLC, which is what I've indicated here with the asterisk. This is the most common defect in these pathways. This protein, which kind of functions as an enzyme, catalyzes the removal of B12 functional groups or moieties, such as the adenosyl group from adenosylcobalamin or the methyl group from methylcobalamin or the cyanide group from cyanocobalamin. Occasionally, this has been overcome by supraphysiologic dosing, specifically of adenosylcobalamin, in cases non-responsive to hydroxycobalamin. So remember what I said, the most common in the medical world, the most common solution to problems in these pathways is to administer high-dose hydroxycobalamin. But sometimes that doesn't work, and rarely, very rarely, some of those cases will be responsive to adenosylcobalamin. So last time, I want you to focus on the entry of B12 through this pathway. One of the branches of this pathway is to take B12 into methylcobalamin. Now I'm going to show you the other half of the pathway. And the other half of the pathway is to take this B12 to adenosylcobalamin within the mitochondria. So we use different transport proteins here. Again, CBLD, CBLB, and CBLA, which is the last step in converting B12 into adenosyl B12. Adenosylcobalamin is the cofactor for the enzyme methylmalonyl-CoA mutase, which converts L-methylmalonyl-CoA into succinyl-CoA, which then enters the Krebs cycle of the mitochondria. So again, as you can see by the diagram, you can see the mitochondria here. We are in the mitochondria right now where all this is happening. L-methylmalonyl-CoA is converted by methylmalonyl-CoA mutase, which requires adenosylcobalamin as a cofactor, into succinyl-CoA, which enters the Krebs or the citrate cycle and then gets basically burnt off as a fuel. This enzyme is only active against L-methylmalonyl-CoA. The other form is D-methylmalonyl-CoA, and I'll talk about that in just a moment. But again, this enzyme that requires adenosylcobalamin is only active against L-methylmalonyl-CoA. Well, where does that come from? This comes from propionyl-CoA carboxylase and its cofactor, which is biotin, which converts propionyl-CoA and propionic acid, which has various sources. So you could think of this as kind of the fountainhead, or this is where things start. So let's kind of refresh our brains really quickly. Here we were talking about B12 metabolism, and now we're talking about why does this matter? And I'm about to explain to you why this matters. So, propionyl-CoA, which kind of interconverts with propionic acid, as you can see written here. Propionyl-CoA comes from gut anaerobic bacterial fermentation of carbohydrates. So propionyl-CoA and propionic acid are produced within your gastrointestinal tract from bacterial fermentation of carbohydrates. You can reduce the production of propionyl-CoA and propionic acid with either fasting to reduce the carbohydrate intake or with an antibiotic such as metronidazole. Propionyl-CoA is also produced metabolically from amino acids such as valine, methionine, isoleucine, threonine, and also odd-chain fatty acids. So propionyl-CoA and propionic acid are converted by this enzyme, propionyl-CoA carboxylase, which requires biotin, into D-methylmalonyl-CoA. So let me say that one more time, perhaps with fewer words. So we go from propionyl-CoA, also propionic acid. We're going to convert those with this enzyme, propionyl-CoA carboxylase, into something called D-methylmalonyl-CoA, which is the same as L-methylmalonyl-CoA, except it's the other isomer. So commonly this is described as the left hand or the right hand. The D form is the right hand and the L is the left hand. The D-methylmalonyl-CoA has to get converted by an enzyme, which I've just abbreviated right here, called MCEE. It has to get converted from the D form to the L form so that this enzyme can act on it. However, in states of inflammation, the cytokine tumor necrosis factor alpha reduces the expression of this enzyme, causing accumulation of the D isomer of methylmalonyl-CoA. That cannot be converted into succinyl-CoA to then get used in the Krebs cycle, and it goes straight to methylmalonic acid, which is what I have right here. Methylmalonic acid is neuroinflammatory, it promotes mitochondrial dysfunction, and it also promotes tumor growth. So that's what I talked about. If this is the part 5 video, then I talked about that in the part 4 video, that methylmalonic acid is not simply an inactive intermediate waiting around for you to test it in a laboratory sample. Methylmalonic acid is directly toxic because it is directly neuroinflammatory, mitodisruptive, as I stated here. It promotes mitochondrial dysfunction and it promotes tumor growth. So if a person has too much inflammation in their body and they're releasing tumor necrosis factor alpha, that's going to reduce the production of this enzyme, MCEE, which converts the D form to the L form of methylmalonyl-CoA. If it remains in the D form, then it's going to create more methylmalonic acid, which is neuroinflammatory, mitodisruptive, and tumorigenic, otherwise known as carcinogenic. So we need to reduce inflammation in order for this conversion from propionyl-CoA through D-methylmalonyl-CoA to L-methylmalonyl-CoA, so that then this can get cleared by this enzyme, methylmalonyl-CoA mutase, which requires vitamin B12. So hopefully all that's clear. Again, this diagram took some work, as you can expect, but at least now I think I've got it to the point where I want it. If we don't have enough biotin, then this enzyme is not going to work, and people will accumulate propionic acid and propionyl-CoA. Accumulation of propionyl-CoA causes dermatitis and neuronal demyelination via inappropriate incorporation of intermediates into skin and nerve membranes, as propionyl-CoA replaces or outcompetes acetyl-CoA and malonyl-CoA in lipid synthesis. So again, for this pathway to work, you have to have biotin and you have to have inflammation under control so that the propionyl-CoA carboxylase can convert the propionyl-CoA into L-methylmalonyl-CoA and then have that get converted into succinyl-CoA and burned off in the Krebs cycle. If this cofactor is not available as adenosylcobalamin, then this methylmalonyl-CoA is going to get converted into methylmalonic acid. If the patient has biotin but they have too much inflammation, so let's say they have the cofactors but they have too much inflammation, then because of their increased expression of tumor necrosis factor alpha, that's going to reduce the expression of this MCEE enzyme, which is going to lead to the accumulation of the D form, the D isomer of methylmalonyl-CoA, and that can only go into methylmalonic acid. So what we need is biotin, controlled inflammation, or low levels, the lowest level possible of inflammation, and we need B12 in the form of adenosylcobalamin so that this propionyl-CoA and propionic acid gets converted here into L-methylmalonyl-CoA and then ultimately into succinyl-CoA and burned off. If it doesn't get burned off by this pathway, the methylmalonic acid is going to accumulate and cause neuroinflammation, mitochondrial dysfunction, and tumor growth. If we never make it this far because the patient doesn't have enough biotin, or if they have a mutation in this enzyme, then they're going to accumulate propionyl-CoA. That's going to lead to dermatitis and neuronal demyelination, as these unusual or semi-toxic fatty acids get incorporated into the skin, which leads to dermatitis, or into nerve cell membranes, which causes demyelination. So hopefully all of that's clear. You can watch the video again, of course, if you need to, but I've covered now the major points of B12 metabolism and why it's important. So you can also understand the different perspective that I offered, which is to think of this in terms of metabolic detoxification. You have to get rid of propionyl-CoA and propionic acid. How do you get rid of it? You have to have a functioning enzyme with the cofactor, which is biotin, so then you can you can convert propionyl-CoA and propionic acid into L-methylmalonyl-CoA. If you have biotin but a lot of inflammation, then the person's going to end up converting this propionyl-CoA and propionic acid into the D isomer of methylmalonic acid, and that's neuroinflammatory, mitodisruptive, and tumorigenic. So we have to get rid of this, otherwise when it accumulates, that's going to lead to metabolic toxicity from the methylmalonic acid. So again, you can measure methylmalonic acid in the blood, just like you can measure homocysteine. I don't think you need to do that if you are suspicious of just a uncomplicated B12 deficiency. But if you're working obviously with a young child who's got failure to thrive, this is a pathway that you would test. And if you're working with a more complicated case that could be B12 deficiency, let's say you're working with an autistic child or an autistic adult or an adult who's got schizophrenia, then you would want some of these insights in order to help guide not only the type of treatment, but kind of the level of aggressiveness of your treatment. And, do you need to go to intramuscular injections or subcutaneous injections? Do you need to try adenosylcobalamin instead of hydroxycobalamin if the defect is in this direction? So originally, as I stated from the beginning in part 1 of this series on B12 pharmacology, I was against using or over-relying on these lab tests, but now I have a slightly different perspective. If it's a simple B12 deficiency, I don't think you need to run a methylmalonic acid test. But if it's a complicated case, whether it's adult or child, or if you want to look at these from a preventive standpoint, like cancer prevention or cardiovascular disease prevention, now I think I have better clarity as to when I would perform these tests, again, whether it's methylmalonic acid or homocysteine. Those tests used to be quite expensive. Now, especially if you work with a laboratory co-op, then you can get those tests for approximately $70. I'm very clear that they used to cost $200 or $300. Now the price has come down, so that's less of a barrier. But again, if you're going to run the test, you need to understand why you're running the test. And I think this diagram that I've now reviewed a couple times, as it has evolved over the last couple of days, I think this diagram helps us to have more clarity on that. So thank you very much. Hope you're all doing well. And I'll keep working on this. I think we'll take one more step with this topic of B12 pharmacology. I think I'll do a part 6, and that's just going to be a formal document that you can download, and it'll have a few more details, but obviously it'll contain this diagram as well. So that brings us to the end of today's quick video. That was as fast as I could make it, even though it turned out to still be almost 30 minutes. So again, I just want to say thank you to all of you who support this work, because it does take time. I mean, this takes hours of work to read the articles, to update the diagrams, to record the videos, to edit the videos, process the videos, and then upload the videos. All this takes a considerable amount of effort, so I appreciate those of you who support it. Thanks, and take care. Bye.
I'm still whooping all throughout the town.